Today I will show you one important structure on the base of the brain, that is interpeduncular fossa. This fossa is an important short note in theory exam, and also in Viva voce examination we also sometimes ask show the interpeduncular fossa and its boundaries and what are the contents of this fossa. Before showing in the specimen, I am drawing this boundary of interpeduncular fossa and the contents, then I will show the specimen. Let us see. Suppose this is the eyeball, and from there you all know the optic nerve coming out and they cross at one region forming the optic chiasma. So this is optic chiasma, and this one is the optic tract, and this is optic nerve, and this optic tract going like this to the superior colliculi of the midbrain, and if this is the pons, this is pons, this is upper border of the pons, this one, and from there this is the crus cerebri of the midbrain from both sides. And here another structure is there, that is olfactory bulb, then it divides into trigon like lateral and medial olfactory stria. This is the olfactory trigon, this one called olfactory trigon, and this is olfactory stria, medial olfactory stria, and this one is lateral olfactory stria. And here is some gray matter called anterior perforated substance, and here you will get one elevation, what is called tuber cinerium with infundibulum, and posterior to it two rounded elevation on each side called mamillary body. Mamillary body, these two, they are the nucleus of the hypothalamus, and this tuber cinerium and infundibulum, it is attached with the pituitary gland, and here in this region you will get again some neurons or gray matter. It is called posterior perforated substance. Now, which one is the interpeduncular fossa? This diamond shaped area, this one is the interpeduncular fossa. Then what is the boundary? Very simple, anteriorly optic chiasma, posteriorly upper border of the pons, anterolaterally by the optic tract, and posterolaterally by the crus cerebri of the midbrain. These are crus cerebri. So this is the boundary, and here the contents are from anterior to posterior. It is tuber cinerium in the midline, two mamillary body, and posteriorly here is a posterior perforated substance. Is a gray matter which is perforated by the central branches of the posterior cerebral artery, and here is the posterior cerebral artery. This is the posterior cerebral artery. From this artery, the central branches enter into this posterior perforated substance, and here is the anterior perforated substance in the olfactory trigon. It is lateral to the optic chiasma and the optic tract, and both sides you will get this anterior perforated substance, and they are interconnected by anterior commissure. And here the gray matter, it is perforated by the branches of anterior and middle cerebral artery, and here is the branches from the posterior cerebral artery. So this is the boundary and content of the interpeduncular fossa in the base of the brain. Now I am showing you in the specimen the boundary and content of interpeduncular fossa. Let's see the specimen. I will show you the boundaries and content of interpeduncular fossa. This is the cerebrum as a whole. This is the upper surface, lateral surface, and this is the base of the brain or under surface. And here we will see the interpeduncular fossa. These two are the temporal lobe and temporal pole. Just I am separating the temporal lobes sideways, and here you can see this is the optic chiasma. Is some part of the optic chiasma is torn here. If you see closely, this is my fossa is holding the area of optic chiasma, this one, and this is the optic tract of this side and optic tract of this side, this one, and this is the crus cerebri of this side and crus cerebri of this side, and this is upper border of the pons. This is the pons, the upper border. So boundary anteriorly optic chiasma, anterolaterally optic tract of both sides, posterolaterally crus cerebri of midbrain of both sides, and posteriorly is the upper border of the pons. This is the boundary. And content from anterior to posterior, here is the tuber cinerium, here this one, and behind it bilaterally the two rounded elevation called mamillary bodies. And in this triangular depressed area, here you will get the grey matter called posterior perforated substance, which is perforated by the central branches of posterior cerebral artery. This is a basilar groove. So this is posterior cerebral artery. From there, the branches enter into this posterior perforated substance. And here, lateral to this olfactory tract, this area is the perforated area. Can you see the perforations? Small openings here. If you closely observe this area, you will see the perforations. These are small perforations. So this area is the anterior perforated substance lateral to this optic tract, and this anterior perforated substance, it is perforated by the middle and anterior cerebral arteries. So this is all about the boundary and content of interpeduncular fossa, and this fossa is on the base of the brain. Above this fossa is the third ventricle, and below is the interpeduncular cistern. So if the question comes on the short note of interpeduncular fossa, you will write down the location or position of the interpeduncular fossa, then draw a diagram and label all the boundaries of the fossa and the contents of the fossa. So that's all for interpeduncular fossa. Thank you.